All right, guys, this is lecture number 2 in our preparation for our biotechnology transformation lab. What we want to do in this lecture is to learn about what transformation is, understand different kinds of transformation that occur naturally, understand also the kind of transformation that we're going to perform in our lab, and then finally understand some of the concepts behind the transformation that we will perform. Because it's important to understand how and why we're doing what we're doing so we can understand the results that we see in order to draw appropriate conclusions. First things first, though, transformation by and large, generally, is simply when the genotype and usually the phenotype of a prokaryotic cell are altered by the uptake of foreign DNA from its surroundings. Ultimately, what that means is a bacterium takes up somebody else's DNA and incorporates it into their own genome, and oftentimes expresses that gene or that genetic information. Transformation in let's say, the natural world can happen in a number of ways. One of those ways in which bacteria can take in and express foreign DNA is through a process known as transduction. All right, I'm going to change the width of my pen. Okay, transduction utilizes what we're going to call a bacteriophage. Ultimately, a bacteriophage is a virus. All right, it's a virus, though, that specifically targets bacteria. Sometimes it's also just known as a phage or a phage. It can be pronounced either way. This phage ultimately binds to the host bacterium, and the phage injects its DNA. The DNA is viral DNA that codes for all of the proteins that make up the phage itself, that allow it to reproduce. The phage DNA and the enzymes that it codes for degrade the viral DNA into fragments. What happens then is the phage then reproduces itself inside the bacterium. And inside that bacterium, the phage reproduces and reconstructs itself and duplicates. It synthesizes new phages that incorporate its own DNA, but sometimes, and importantly, mistakenly, some of that bacterial DNA that had been put into fragments. So this particular phage right here possesses some of the bacterial DNA along with its own phage DNA. And that phage now can attack and insert new DNA into a new bacterium. Of course, the DNA that it's now inserting is DNA of the virus itself, but also of the bacteria, the original host bacteria. So now what we've done is we've inserted bacterial DNA, new foreign bacterial DNA, into a new bacterium cell. Sometimes that bacterial DNA can then reincorporate itself into the host genome, and then can be expressed in many ways. That cell is then said to be transformed. It's a recombinant bacterium. So through the use of a bacteriophage, bacterial DNA can be inserted and incorporated into some new bacterium. Of course, if that gene is different, if it has a different allele than the second host, then there will be some recombination and a transformation of phenotype. Another way in which bacteria can be transformed is through a process known as conjugation. In conjugation, specifically with what we're going to call the F plasmid, we're using a plasmid. This F plasmid codes for what we're going to call this conjugation pilus, or one of my favorite terms and favorite ideas in all of biology. It's also known not as a conjugation pilus, but as a sex pilus. And so this sex pilus ultimately creates this kind of channel or a connection point between two bacterial cells. And through this channel then, what you can see is we've created a channel, and the original bacterial cell that has this plasmid, that has this F plasmid, we're going to call it the F plus cell. This F plus cell sends the plasmid through the sex pilus, or the conjugation pilus, to an F minus cell, or a cell that does not have the F plasmid. What happens then is, as you can see, there's a replication going on that becomes double-stranded, and now ultimately we've got two new cells that both have the F plasmid. Both cells are now F plus. Both cells have new DNA, and this cell right over here, this second cell, not the newest F plus cell, is said to be transformed. Here we can see an image, an actual sort of microscopic image of two bacterial cells and the sex pilus. And through this sex pilus, of course, one bacterial cell is going to be sending genetic information, whether it's through a plasmid or, as we'll see next, through the F factor that had been incorporated into its own genome. If by chance this F factor in the F plasmid becomes incorporated into the bacterial genome, it's now considered an HFR cell called high frequency of recombination. This HFR cell can act in many ways very similarly to any other F plus cell that has an F plasmid. Of course, it doesn't have a plasmid. So what happens is, through the sex pilus, the donor cell, or the F plus cell, can sort of snip its own genome and send the plasmid through, send the F factor through. As the F factor is sent through, it's being replicated and turned into double-stranded DNA. And what happens, of course, is the entire genome is not sent through. Only part of it, an incomplete F plasmid, is sent through. What will happen then is we've got the F factor here. Now we have extra DNA. We have extra genes, extra genetic material that had been sent into this new cell. So this new bacterium having extra genes, these genes can be recombined through crossing over into the original genome. Again, that would be another form of transformation. The three forms of transformation we just looked at, both transduction, conjugation, and conjugation through an HFR cell, those are kinds of transformations that occur in the natural world. We're going to sort of kind of copy that. We're going to sort of simulate that, but it's going to be a little different. Instead of having two bacterial cells sort of communicating and sending information to one another, we're going to sort of artificially incorporate a recombinant plasmid into a host cell. We're going to be using specifically the bacterium E. coli. And what we're going to be doing is we're going to be, to this E. coli, we're going to add what we're going to call heat shock. We're going to add some heat shock. A heat shock, if I can spell it correctly, what we do is we expose it to some cold environment, then we immediately put it in something that's quite warm, and then we bring it back into a cold environment. What this does is it creates a bacterial membrane that is easy to pass through. We actually create a more permeable bacterial membrane. Of course, a more permeable bacterial membrane will be easier for our recombinant plasmid to enter into our cell, thus transforming it. The specific plasmid that we're going to be using is a plasmid that has a few genes on it. But before we talk about those genes, it's also important to know that that this plasmid has enough promoters and operons such that the cellular machinery can transcribe and translate this plasmid just simply by being in the bacterial cell. We don't have to incorporate it into the bacterial genome. Alright, so what we see in this plasmid is, first of all, we see this space. All right, that's going to be the origin of replication. That's ultimately this space in which whenever the plasmid is replicated, it always starts at the origin. That's not that important. That's not that vital to what we're looking at. But what we are looking at, however, is this gene. It's a gene for beta-lactamase. And beta-lactamase, as an enzyme, what it does is it inactivates ampicillin, an antibiotic. So therefore, if this protein inactivates ampicillin, it creates an ampicillin resistance for the bacteria that have this plasmid. That's going to be really important. The other thing that's going to be really important is this gene. It's a gene that creates a regulatory protein, but it's a gene that responds to arabinose. Arabinose is another sugar. And so when arabinose is present, we have a regulatory protein. We're going to call it protein C. When protein C is translated, it will attach to another promoter, this promoter here, and it will code for another protein called the green fluorescent protein. The green fluorescent protein is simply a protein that gives the property of fluorescence, and specifically a green fluorescence. Thus, when a bacterial cell incorporates this plasmid, it has several important characteristics. Number one, it is resistant to ampicillin. Number two, when arabinose is present, arabinose will activate through the operon this regulatory protein C. When regulatory protein C is transcribed and translated, it activates this gene for the green fluorescent protein. Therefore, if bacteria have taken up this plasmid, they will be resistant to ampicillin, and they will fluoresce green. This diagram is a quick rundown of the procedure that we'll be using. I encourage you to pause this video, take a look at this, and understand what we're going to be doing, as well as reading the procedure in your handout. So that about wraps up our discussion of what transformation is and some of the context and the contextualized concepts that you're going to need to know in order to understand what and why we're doing it in our lab. What's next, however, I'd like you, after you listen to this lecture, I'm going to be giving you a link to a Google form, and you'll be filling out that Google form with information that comes from one of the handouts. It's actually a teacher and student manual from the people who created the lab. It's a big handout, and I'm going to direct you to certain areas of that handout and ask you a few questions about, number one, sterile technique, and number two, some of the conceptual ideas that we're dealing with, specifically how many colonies that you'll need to scrape from your agar plates. What should your inoculating loop look like? What is an inoculating loop? What is heat shock? Why do we do it? And how do you do it? And specifically, why we're adding ampicillin? Why is that important? Why are we heat shocking? And why are we adding arabinose? All right, so stay tuned for that, and email me if you have any questions. This is a quick diagram that indicates actually the three kinds of agar plates that I've created and that we'll be using in our lab on Friday. Let's go from right to left. The first one is simply an agar plate that has LB nutrient broth. LB stands for the two names of the people who developed this nutrient broth. It has nutrients that allow bacteria to grow, and it has agar to create some kind of gelatinous consistency so that bacteria can grow on top of it. The next agar or nutrient plate that we'll be using is one that has the LB nutrient broth as well as ampicillin. And so, of course, the bacteria that have incorporated the plasmid will be able to grow on this. The bacteria that have not incorporated the plasmid will not be able to grow on this plate. And so this will be our control. Finally, the third type of nutrient agar plate is one that has the LB broth, it has ampicillin, and it has arabinose. And so if there's arabinose, then the regulatory protein C will be activated, and thus activating the the green fluorescent protein, the GFP. And so these should be the ones that the bacteria that have incorporated the plasmid should be fluorescing green.